Anxiety and fear are not signs of weakness. They are signals. Signals that your brain and nervous system are doing exactly what they're designed to do. Protect you. But what if I told you that the same biology that creates anxiety can also be trained to overcome it? Today, I want to share with you how neuroscience, not guesswork, not vague advice, gives us the tools to rewire our brain, calm our nervous system, and take back control of our thoughts, our emotions, and ultimately our lives. Either great pain. I'm going to break down the science of how fear is formed, where anxiety lives in the brain, and the exact steps grounded in real pain, peer-reviewed neuroscience, that you can take to shift your brain from a state of fear and paralysis to clarity, action, and confidence. Because understanding your brain is the first step to mastering it. And when you master it, fear becomes fuel and anxiety becomes awareness. The biology of fear begins in the amygdala, great pain, a small almond shaped structure deep in the temporal lobe that acts like an early warning radar system for threat detection as soon as sensory information, sights, sounds, bodily sensations, passes through the thalamus, great pain. A rapid, low road projection delivers a stripped down version of that data straight to the amygdala, allowing it to initiate protective responses even before conscious awareness forms. In parallel, a slower, high road sends the same information to the sensory cortices and onward to the prefrontal cortex, either great pain, where richer analysis occurs. When the amygdala senses danger, it triggers a cascade through the hypothalamus, great pain, activating the sympathetic branch of the autonomic nervous system. Heart rate accelerates, pupils dilate, and adrenaline floods the bloodstream, priming muscles for action. Simultaneously, either great pain, the amygdala signals to the periocaudal gray to initiate defensive postures, while the insula heightens interoceptive awareness, making you acutely conscious of your pounding chest or tightening gut. All of this happens in a fraction of a second, great pain, creating the subjective feeling of fear long before rational thought weighs in. Yet the prefrontal cortex, especially the ventromedial and dorsolateral sectors, has the capacity to reinterpret or regulate the amygdala's alarm. Through reciprocal inhibitory pathways, the prefrontal cortex can release GABAergic signals that dampen amygdala excitability, essentially telling the brain, I've assessed this, we're safe. Functional imaging studies reveal that individuals who practice cognitive reappraisal, consciously reframing a perceived threat, show increased prefrontal activity coupled with decreased amygdala activation, illustrating this top-down control. Yeah. Chronic stress or early life trauma can bias the system toward hyperreactivity, great pain, strengthening synaptic connections from the amygdala to the brainstem while weakening prefrontal regulatory circuits. Cortisol, released during prolonged stress, can even shrink dendritic arborization in the prefrontal cortex, reducing its capacity to modulate fear responses. Meanwhile, the amygdala's dendrites thicken, making it more sensitive. This explains why anxious brains feel hijacked. The hardware literally tilts the balance toward threat detection. Importantly, neuroplasticity works both ways. Pain, repeated activation of prefrontal circuituses through practice like mindfulness meditation, cognitive behavioral therapy, or structured exposure rewires synapses, enhancing the inhibitory control over the amygdala. Rodent studies show that consistent exogen training, exposing an animal to a conditioned stimulus without the aversive outcome, strengthens infralimbic cortex pathways that suppress amygdala neurons. Human fMRI parallels confirm that deliberate graded confrontation with feared cues grows prefrontal amygdala connectivity, allowing the brain to recategorize the stimulus as non-threatening. Breathwork provides a rapid bottom-up route. Slow exhalation, biased breathing activates vagal afferents that project to the nucleus of the solitary tract and onward to the locus coeruleus, reducing noradrenaline output and calming the amygdala indirectly. Likewise, specific eye movements, such as widening the visual field to panoramic vision, shift activity from the amygdala to the parietal cortex, signaling safety by mimicking the relaxed scanning state used in open landscapes. Even posture feeds into the circuit. Upright, expanded stances send proprioceptive feedback through spinal pathways that reduce limbic arousal. Layered together, these insights reveal that fear is not a monolithic emotion, but a dynamic dialogue between subcortical alarm systems and cortical control centers. By understanding how the amygdala and prefrontal cortex interact and how hormones, neurotransmitters, and bodily feedback shape that interaction, we gain leverage points for intervention. Each conscious reframe, each slow breath, each exposure repetition is a vote cast for the prefrontal cortex, teaching the brain that what was once interpreted as danger can, through experience and restructuring, be reclassified as manageable challenge, thus converting the raw signal of fear into a catalyst for adaptive action. The autonomic nervous system, operating far below conscious awareness, orchestrates nearly every physiological change that accompanies anxiety and calm alike. Its two primary branches, the sympathetic and the parasympathetic, function less like simple on-off switches and more like dials that can be tuned moment to moment. When the sympathetic branch surges, driven by the release of catecholamines such as norepinephrine from the locus coeruleus and epinephrine from the adrenal medulla, heart rate climbs, bronchial passages widen, blood pressure rises, and glucose floods the bloodstream, mobilizing energy for rapid response. Simultaneously, blood is shunted away from the digestive tract toward skeletal muscles, while pupils dilate to sharpen visual acuity, all of which add up to the distinctly visceral sensation we label as anxiety. Yet this sympathetic dominance is never absolute. The parasympathetic branch, anchored by the vagus nerve, remains present, ready to counterbalance the arousal by slowing cardiac pacemaker cells, stimulating digestive enzymes, and releasing acetylcholine that dampens inflammatory pathways. The dynamic interplay between these branches determines how intensely we feel anxiety and how swiftly we can recover. Heart rate variability offers a window into this dance. Higher variability indicates a flexible autonomic system capable of shifting from vigilance to relaxation with agility. Studies show that individuals who practice slow, diaphragmatic breathing at roughly 6 breaths per minute exhibit pronounced oscillations in heart rate variability, reflecting strong vagal modulation of the sinoatrial node. These breathing induced vagal bursts synchronize with baroreceptor feedback loops in the aortic arch and carotid sinus, sending afferent signals to the nucleus tractus solitarius, which in turn projects to the ventrolateral medulla to modulate sympathetic outflow. In effect, each deliberate exhale lengthens the interval between heartbeats, creating a micro recovery period that, when repeated over minutes, nudges the overall autonomic set point toward calm. Functional MRI studies reveal that such breathing not only influences brainstem nuclei, but also reduces activity in the anterior insula and increases connectivity between the medial prefrontal cortex and the amygdala, providing top-down reassurance that the body is safe. Beyond breath, posture and movement feed directly into autonomic circuits. When you stand tall with shoulders, back, and head elevated, stretch receptors in spinal musculature send proprioceptive input via the dorsal column to the somatosensory cortex and cerebellum, signaling stability and readiness rather than collapse. This afferent feedback biases the hypothalamus toward parasympathetic tone. Likewise, rhythmic physical activities, walking, cycling, swimming, generate regular afferent signals through mechanoreceptors that entrain brain stem pattern generators, indirectly reducing sympathetic noise. This explains why even a 5-minute brisk walk can disrupt a spiraling panic episode, not merely through distraction, but through tangible shifts in autonomic balance. Nutrition further modulates this system. Foods rich in tryptophan and omega-3 fatty acids support the synthesis of serotonin and promote membrane fluidity in neuronal cells, making vagal firing more efficient. Conversely, high glycemic diets spike insulin and cortisol, tipping the scale toward sympathetic predominance. Hydration also matters. Mild dehydration elevated angiotensin tellier and vasopressin hormones that augment sympathetic tone. Thus, small lifestyle choices, choosing complex carbohydrates, drinking water, supplementing with EPA-DHA, collectively shape the autonomic milieu in which anxiety either thrives or diminishes. Circadian factors overlay this intricate network. Cortisol follows a predictable rhythm, peaking shortly after waking and declining through the day. When sleep is truncated or irregular, the cortisol slope flattens, leaving background sympathetic activity elevated throughout the night. Deep, slow, wave sleep is dominated by parasympathetic processes, allowing cellular repair and glial clearance of metabolites, while rapid eye movement sleep features transient sympathetic spikes that rehearse threat scenarios without physical risk. Chronic sleep restriction truncates both phases, leading to accumulated sympathetic bias and reduced capacity for parasympathetic rebound, which manifests as persistent morning anxiety. Strategic light exposure, bright outdoor light within an hour of waking and dim amber light in the evening, reinforces the suprachiasmatic nucleus signal that synchronizes autonomic rhythms with the external world, thereby strengthening the daily oscillation between arousal and rest. Social connection is another potent modulator. Eye contact, facial expressions, and vocal prosody engage the ventral vagal complex described by polyvagal theory. A genuine smile or a warm tone activates myelinated vagal fibers that slow heart rate and encourage digestive activity. Oxytocin release during supportive interactions further dampens hypothalamic, pituitary, adrenal axis activation, lowering cortisol and enhancing parasympathetic modulation. Even brief, positive social cues, thanking a barista, exchanging a friendly nod, populate the day with micro doses of vagal stimulation that accumulate into measurable reductions in baseline anxiety. Importantly, the autonomic nervous system is trainable at every age. Practices such as resonance breathing, yoga nidra, and mindfulness meditation consistently enlarge the Preussinger complex's ability to generate calm breathing rhythms and reinforce vagal nuclei connections. Over weeks, this rewiring leads to decreased baseline levels of norepinephrine and a recalibrated threshold for sympathetic activation, making everyday stressors feel less like threats and more like manageable challenges. Biofeedback devices that display heart rate variability in real time provide immediate evidence of progress, converting abstract neurophysiology into tangible, gamified scores that motivate persistence. What begins as deliberate practice gradually becomes trait, level resilience as synaptic plasticity cements a new autonomic equilibrium favorable to calm alertness, sharpening cognition without tipping into anxiety. Breath and vision offer two of the fastest levers for steering neural activity because they plug directly into circuits that monitor internal carbon dioxide levels and external spatial cues. By manipulating these inputs, you can push the nervous system toward heightened alertness or deep calm in under a minute. A single deep inhale followed by a longer, slower exhale, a maneuver known as a physiological sigh, rapidly drops sigh all partial pressure in the bloodstream, causing chemoreceptors in the aortic arch and carotid bodies to signal the medullary respiratory centers to slow downstream sympathetic drive. This exhale lengthening also recruits vagal afferents that project to nucleus tractus solitarius, which then dampens locus coeruleus firing, lowering cortical norepinephrine and giving a felt sense of immediate relief. Repeating 2 or 3 of these sign cycles primes the diaphragm to initiate more efficient ventilation patterns and raises heart rate variability, demonstrating real-time parasympathetic dominance. Conversely, short, rapid inhales through the nose without full exhalation raise col, stimulating the retrotrapezoid nucleus and accelerating sympathetic tone. Athletes exploit this to ramp up arousal before competition, proving that breath is a bidirectional throttle rather than a one-way brake. Layering tempo onto this equation matters. Breathing at roughly 6 cycles per minute synchronizes baroreflex oscillations, while resonance patterns nearer to 1 inhale every 4 seconds and 1 exhale every 6 create the greatest amplitude in heart rate variability, a metric tightly linked to stress resilience. Nasal breathing confers bonus neurochemical effects via olfactory bulb activation, sending gamma frequency bursts to hippocampus and piriform cortex that sharpen focus while still supporting calm. Breath holds at peak inhalation stretch pulmonary baroreceptors and briefly elevate blood pressure, triggering compensatory vagal rebound upon exhalation that many people feel as a wave of relaxation cascading down the torso. Beyond chemistry, the mechanical action of the diaphragm massages the vagus nerve as it passes through the esophageal hiatus, providing direct electrical like stimulation akin to implanted vagal stimulators used in clinical anxiety treatment. Vision offers an equally powerful but often overlooked portal into state control. When you narrow your gaze to a tight focal point directly in front of you, the midbrain superior colliculus and parietal eye fields heighten sympathetic arousal, preparing you for goal-directed action by boosting pupil dilation and enhancing contrast sensitivity. This near vision mode raises activity in basal forebrain cholinergic neurons, sharpening attention at the cost of increased limbic tone. In contrast, deliberately expanding your visual field, softening the gaze to take in the periphery, activates wide field motion detectors in the maxillar pathway and shifts processing toward the posterior parietal cortex, signaling safety because predators rarely attack when the horizon is broad and threats are easily spotted from afar. This panoramic vision suppresses amygdala output and correlates with reduced beta power in EEG readings, mirroring meditative states. Horizontal eye movements of about 1 hertz, similar to those used in EMDR therapy, rhythmically shuttle information between hemispheres via the corpus callosum and dampen activity in the anterior cingulate cortex, which is often hyperactive during rumination. Even vertical gaze shifts matter looking slightly upward, engages midbrain dopaminergic circuits associated with motivation and optimism, while consistent downward gaze can correlate with decreased cortical serotonin and a slump in mood. Thus, consciously lifting the eyes to the upper third of the visual field can subtly elevate effect within seconds. Combining breath and vision multiplies their impact because both modalities converge on brainstem hubs that gate autonomic output. For instance, pairing a physiological sigh with an immediate transition to panoramic vision can drop systolic blood pressure by 5 to 8 millimeters of mercury in under 30 seconds, as shown in laboratory biofeedback trials. Conversely, a rapid nasal inhale series coupled with a narrowed forward cast stare primes the reticular activating system, making you more vigilant for complex tasks like public speaking or late night driving. These effects are trainable. Practicing intentional breath vision cycles for 3 minutes twice daily increases baseline parasympathetic tone over a month, measurable through resting heart rate reductions and decreased salivary alpha amylase, a proxy for sympathetic activity. Neuroplastic adaptations include strengthened inhibitory projections from prefrontal cortex to amygdala and enhanced synchronization between insula and anterior cingulate, regions that jointly interpret bodily signals as emotions. On a behavioral level, people report faster recovery from surprise stressors like sudden loud noises because the brain learns to associate specific interoceptive and visual cues with safety. The portability of these tools makes them uniquely empowering. Unlike meditation apps or therapy appointments, your lungs and eyes are always with you, offering an on-board control panel for state regulation that requires no external equipment and very little time, making consistency feasible in busy schedules or high-pressure environments. Neuroplasticity transforms fear from an unyielding reflex into a modifiable memory trace by exploiting the brain's natural ability to reshape synaptic connections. Each time a representation is reactivated, every instance you recall or confront a fear, the associated neural ensemble in the basolateral amygdala and hippocampus becomes temporarily labile, opening a molecular reconsolidation window that lasts for roughly 6 hours and is governed by protein kinase M-zeta, CRE transcription factors, and AMPA receptor trafficking. Intervening during this window with carefully timed repetition, whether through graded exposure therapy, imaginary rehearsal, or even immersive virtual reality, can update the memory with safety signals that overwrite the original threat prediction, weakening the probability that the amygdala will fire the next time the cue appears. Long-term potentiation and long-term depression serve as the cellular currency of this change-frequent, low-intensity exposures induce calcium-dependent LTD, thinning synaptic spines on excitatory neurons that would otherwise amplify the alarm, while occasional high-salience successes, such as staying in a feared environment until anxiety naturally subsides, produce bursts of brain-derived neurotrophic factor that stabilize new inhibitory interneuron synapses projecting from the ventromedial prefrontal cortex. Repetition also recruits the dorsal striatum's habit circuitry. After dozens of approach-avoid cycles, the act of stepping toward a previously feared stimulus becomes procedural rather than deliberative, offloading cognitive effort and freeing working memory for nuanced problem solving. Sleep slices this process into clean, reinforcing segments during non-REM stage 2 thalamocortical spindles replay exposure sessions, consolidating declarative aspects of the learning, while REM episodes selectively downscale amygdala synapses that failed to elicit danger, effectively pruning obsolete fear pathways through synaptic homeostasis. Pharmacologic adjuncts can amplify repetition's effects. A single 50 milligram dose of D-cycloserine administered 1 hour before exposure boosts NMDA receptor-mediated plasticity, doubling the speed at which extinction memories dominate. Conversely, a propranolol given immediately after reactivation blocks noradrenergic consolidation of the original fear trace, trimming its emotional intensity without erasing factual content. On the behavioral side, grain variability within repetition matters. Alternating context, time of day, or sensory cues during exposure broadens the hippocampal indexing of safety, preventing renewal when conditions change. This diversity encourages the dentate gyrus to form pattern, separated engrams, so that the broader concept of elevator or crowded street feels neutral rather than just the one specific elevator used in therapy. Great pain or mental imagery repetitions, though less potent than real-world exposures, still drive measurable shifts in functional connectivity between the anterior insula and temporoparietal junction pain, regions that translate bodily sensations into conscious feelings, combining imagery with interoceptive awareness, like noticing the exact texture of the heartbeat while picturing the feared situation, accelerates reclassification by teaching the predictive coding network that sympathetic arousal need not equal catastrophe. Great pain, even micro repetitions throughout the day, accumulating a 5-second glance at a photo of a spider followed by a physiological sigh and a reframing statement may seem trivial, but across weeks it adds hundreds of reconsolidation events, each nudging synaptic weights downward in the threat circuit. The wearable devices now allow immediate capture of those micro events. Pressing a tactile button whenever a fear cue is encountered triggers haptic feedback and logs the instance, providing data-driven proof that exposures are increasing and anxiety spikes are shrinking, which itself reinforces motivation via the mesolimbic dopamine pathway. Social repetition layers still more plasticity, practicing approach behaviors in front of empathetic activates the anterior cingulates social pain circuitry in a safe context, either great pain, teaching the brain that vulnerability followed by acceptance is survivable pain, thus replacing isolation-driven cortisol surges with oxytocin-mediated calm. Crucially, repetition must straddle the sweet spot of intensity known as the learning zone, where arousal is sufficient to trigger plasticity, but not so high that it impairs prefrontal regulation. Eat pain. Tools like subjective units of distress scales and real heart rate monitoring keep exposures inside this band, maxing either maximizing LTP in cortical safety pathways while minimizing amygdala sensitization. Over months, structural MRI scans reveal increased myelination of uncinate fasciculus fibers linking the orbitofrontal cortex to the amygdala grain, a white matter upgrade that speeds top-down inhibitory signaling. Parallel diffusion tensor imaging shows decreased fractional anisotropy in fear-generating subcortical tracts, indicating less rigid synchronization. The behavioral payoff surfaces in metrics such as reduced startle reflex amplitude, longer behavioral approach times, and improved scores on the Leibowitz social anxiety scale. Eat pain, each corroborating the underlying synaptic edits wrought by thousands of tiny, intentional repetitions that, taken together, great pain, reforged the nervous system's predictive models of safety and threat